Yeah, actually, just a minute. Is it hold on? So,
Yes, sir. Yes, sir.
is it okay?
It's visible, sir.
Yes, sir, it's visible.
Yes, sir. So we shall be starting right now. So all of you, very good evening to all of you participants from different colleges. This is so Dr. Biswajit Mukherjee from Jadavpur University doesn't need any introductions. He is a legend in the field of pharmaceutics and in the field of the site-targeted drug delivery. He was my mentor in my PhD, and he was a strong staircase which has led to achieve my PhD and my PDF degree also. So he is a living legend in pharmaceutics from Jadavpur University. Not a single person in India is there who doesn't know his name. He's a very, very famous person in his area, and I will just have the honor to describe his CV in a few words to all of you. So, Dr. Biswajit Mukherjee is a PhD and a professor in pharmaceutics in the Department of Pharmaceutical Technology, Jadavpur University. He's a former JAD scientist, that is the German Academic Exchange Service. He's the coordinator for the AICT sponsored QIP nodal cell in the Department of Pharmaceutical Technology in JU, and he's also a joint coordinator center for advanced research in pharmaceutical technology, JU. In addition, he is the convener of the Animal Ethics Committee of JU, and he is the coordinator of Department of Science and Technology, DST India, in the PERS Program 2 of the Department of Pharmaceutical Technology, JU. He is a former visiting fellow of the Indo-Hungarian Education Exchange Fellow, a National Research Institute for Radiobiology and Radio Hygiene, which is in Budapest, Hungary. He is a recipient of the Finland Government Scholarship. He has worked as a guest scientist in German Cancer Research Center Heidelberg after receiving the Overseas Research Associate Award from Department of Biotechnology, DVT, Government of India. He was also a former faculty in the UIPS, that is the University Institute of Pharmaceutical Science, that is in Punjab University, Chandigarh. He worked as a junior executive in UP Drugs and Pharmaceutical Company Limited in Lucknow. He works on antisense technology and the chemoprevention in cancer model. He has been working on the novel drug deliveries, especially about the transdermal patches and the nanosized liposomal and the neosomal formulations as well as nanoparticles. He has supervised many projects of different funding agencies like as the UGC, CSIR, then ICMR, DST, DBT, DRDO, and he has guided number of PhD scholars and the master degree students. He has published his work in many international journals of repute. He has also written a few books and the book chapters, and a lot of patents are in he is having in his kitty. He's in the editorial board of some international journals of repute. He is the recipient of the UGC Research Award 2009, and along with a lot of other central government awards. He is acting as the editorial board member, editor, and the guest editor for many international journals, and he was a section editor of Encyclopedia of Metalloproteins by Springer. He became the co-chairman, Scientific Service Committee, LOC, 56 Indian Pharmaceutical Congress in Calcutta, India, in the first week of December 2004. He is the Honorary Secretary of Indian Association of Pharmaceutical Scientists and Technologists, Calcutta, India. He has participated in different seminars and conferences to deliver invited lectures both in India and in abroad. He delivered annual symposium of the logo of the double IC, that is the Institution Innovation Council, which is coming under Ministry of Education, Government of India, and it is a joint collaboration with our IQAC cell from our Department of Media Arts. So over to you, sir.
So, first of all, I like to convey my sincere gratitude and thanks to the PG Medical PG Institute, I think PG Institute. Rather, we are working with the carrier of the drug. That means to delivery system of the drug. So, like you know that you are taking paracetamol, the vehicle delivery system is tablet. You are taking sometimes some vitamin solutions, that the delivery vehicle is the liquid. Sometimes delivery vehicle is the capsules. Sometimes delivery vehicle is the other means like aerosols. So here the delivery vehicle of the drug is nano size formulation. So nano size is typically, you can see tablets, you can see capsules, you can see liquid formulation, you can see many of these formulations, powder formulation, but it is very difficult to see the nano formulation. Nano formulation means the size is very, very small. You understand? It is sub-micron level. Sub-micron level materials, you cannot see it by naked eyes. So it is always very difficult that what you are working with, whether you are performing in a correct way or not, until and unless after the production, after you are just evaluating it by a very sophisticated kind of devices. Now, not only that, it is just simply not the development of nano formulation, but you are actually using a ligand or some other method by which you are targeting the formulation to the cells. You see, when we are consuming a drug, it is getting distributed throughout our body. But it is not needed. Actually, basically, it is not needed that drug should be distributed throughout our body. It should be distributed to the location where we have the sufferings. But what happened, we have no such precise formulation so that we can exactly deliver the drug to the right location in the system. I'm not talking about non systemic drug. Like you have an ulcer on the hand or some infection on the leg, you can take the medicine and just apply on it. You don't need to apply from head to the toe. Just you're applying on that particular location. This is the topical, external. But when you're taking internal, that is your infection is there in stomach or maybe in intestine or maybe in liver or some problem or is there in pancreas. You cannot just deliver the drug exactly to that location. It will be distributed throughout our body. So that causes, unnecessary requirement of more drugs. More drugs are not your food. Drugs are not very friendly to you. They, if you consume, there is a continuous sound from the other side. Maybe people are joining. Can you just stop that sound? It will be nice for me. So, what happened? What I am telling you that yes, when you are just or you can locate the drug, you can bring the drug to the right location. You don't need to give lots of chemicals which unnecessarily cause toxicity. So basically, targeting is very important. Many drugs are very toxic. Many drugs are substantially toxic for the system. So it is very important for those drugs to deliver at the right location. Otherwise, you have to consume more. And you know that if you consume it more, the body has normal cells, normal regular cells. They will also get affected with these drugs and they will suffer from toxicity. That's the thing. So basically, we use targeting for that. That means to bring or to deliver the drug at the right location. In a simple way, you want to go to New Delhi, but you are going to Bombay, Chennai, Bangalore, Hyderabad, then in yeah, Guwahati, Agartala, then maybe Darjeeling, and then suffering will be there. Same thing is happening when you are just unable to deliver the drug to the right location. You are going to somewhere else. You are supposed to go to New Delhi. You are going to the other places, and also New Delhi. So it costs lot more in terms of financial point of view for you. Similarly, in body, it costs lot in terms of the toxicity problem. So basically, at this moment, what I want to discuss nanoformulation, and with this nanoformulation, we want to target it. This nano size, as I told you, cannot see it by naked eyes, even not by light microscope. Only some sophisticated instrumentation. More precisely, I can tell you like SEM, scanning electron microscopy, or TAME, transmission electron microscopy. They are the, you can say, equipment by which you can visualize it. You can see it. And also some other instrument like, particle size determination system or data by which you cannot see, but you can measure their size. So basically, the targeting means, I told you, nanoformulation and targeting. Targeting means there are two approaches. One is the conventional approach. Conventional approach means you are just tagging it up with a particular material which helps to bring it to the right location. So that is, we say, conventional approach. Like what you call is, you are conjugating with the antibody, that antibody will bring it to the right cells, those where the specific antigen is getting expressed. So thereby you can say that it will be delivered to the right location. That is called conventional. That means you are using a ligand, you are using a material attaching to the formulation which helps to bring it to the right location. The other is non-conventional way of targeting. Non-conventional, which is you are also targeting the formulation to the right destination, right site of delivery, but here you are not using any ligands. You are using some other method. I will discuss both the things together today so that you can have a view on it. But before that, you have to understand that why we are just going to change the materials into nano size. What is the problem with tablet or micro size formulation, microparticle? Why you are planning to go for nanomaterials, nanoparticle? The reason is that when you are bringing it to the nano size, it gives lots of new properties, additional properties. These new properties, which are quite different than the existing property. This is very, like in macro size, micro size, some properties are there. When you are converting into nano size, you will get absolutely a different kind of property which is not available for macro size or other micro size particles. Example is, sometimes if you bring some materials into nano size, you can make it soluble. Sometimes if you bring it into the nano size, it will have supermagnetism on the surface. Sometimes you are bringing it into small size, what will happen in case of medicinal point of view, or for us, it is very important, the drug which was not penetrating inside the cells, it will go inside the cells. So there are lots of advantages when you are bringing it into the nano size. That is the purpose people prefer to bring it into the nano size. Now I will focus mostly in the area where we can consider to be nanomedicine. That means my focus is just with the nano formulation as a delivery system, not with the other uses of nanomaterial. That nanomaterials has lots of uses, lots of significance. But however, these nanomaterials I will discuss in terms of nanomedicine. Nanomedicine means it has the therapeutic, diagnostic values, and that things we will discuss. Those things, those materials should have the clinical significance, and also it will minimize the toxicity. So I will discuss the toxicity point of view later on at the end of my lecture. Now, if there is this nano formulation, or there are nano formulation, what are the different types of nano formulations available, how we can develop it, etc. So basically, there are several different types of nano formulation. Few of them I have mentioned, like carbon nanotube, protein cage structure, viral nanoparticle. Even you can use the viral code, that's removing the nucleic acid from it. And also, dendrimer, solid lipid nanoparticle, polymeric micelles, and also nanoliposome and polymeric nanoparticle. Basically, I will concentrate my lecture into this area, polymeric nanoparticles and nanoliposome. If you ask me why, because I am working in those two areas. So basically, I will discuss on them. I think this is important. You have studied with this nanoparticle maybe in your classes. You have studied with the nanoliposome in your classes, or maybe in your undergraduate, postgraduate classes. So today you understand that how do they look like exactly, you can visualize them, and also you will come to know something about this. So this is the first work I want to present. These was published in Nanomedicine in 2013. I have mentioned it because this will be helpful for you, those who are willing to work in the area. You can see the work, because in presentation everything cannot be presented. One paper, if I present it, will complete the time. So I am going to present few papers. So naturally, in short, I will present. Basically, what is there? We have developed PLGA nanoparticles, and the drug is your voriconazole. Purpose is pulmonary targeting, pulmonary delivery. That means in lungs. And we have investigated it in terms of in vitro and in vivo. Let me first tell you, in short, you can see the picture. Animals, after the nanoparticles given through nasal route, or oral and nasal, not oral, I should say nasal route, you know, it will go to lungs and it will go to alveoli, and that we have actually observed. See, these are the nanoparticles. You will find the different sizes of nanoparticles are here, different sizes. Some of them are porous also. We have developed porous nanoparticle using certain combination of effervescence combination. So you see, thereby we have made it porous. You can see one at the first, second, third, fourth, fifth. That is under E. You can see the porous nanoparticle, so nanoparticles of different size we have developed, and that they have been, of course, they contain the drug voriconazole. Voriconazole is antifungal drug, and it is purpose is to use them for animal to see whether it is going and reaching to the lungs or not, and how long they're staying there. Let me tell you one very important information in this regard, that why we are trying to deliver the nanoparticles in lungs, or nanoparticles containing voriconazole in lungs. The purpose is, very simple. You know that lungs, normally you will have the fungal infection in lungs when the patient is suffering from different immunocompromised diseases. That is immunocompromised patient. Like patient is suffering from cancer, patient is suffering from diabetes, patient is suffering from HIV, patient is suffering from different hepatic problems, like different hepatitis infection, long-term infection, people are suffering from. So those immunity is very poor. In those cases, we say they are immunocompromised patient. That means the patient has poor immunity. Now, in those cases, actually, the patients having poor immunity, we are just trying to give the drug, antifungal drug, if the person suffers from, fungal infection in lungs. Normally, this compromised patient suffers from fungal infection in lungs. And once they get the fungal infection in lungs, you can say that they have no option but to die. Because we have different, good antifungal drugs like voriconazole, itraconazole. There are many other good antifungal drugs. The problem is not the drug. Problem is, the lungs has a very high and fast blood turnover. So it reaches in lungs very quickly blood, and also it is getting removed from there. So residence time of the drug, you understand residence time, what I mean to say. Residence time means how long it will be in contact with the tissue, because it is very important that it should be absorbed in the tissue. So alveoli, it has a very less tissue mass. It is very empty type, and because they have, what you call the air sac, air is there. So tissue mass is very less. So under that particular circumstances, residence time of the drug is very important to kill the fungus. What happens, that if you give the drug with the usual dose, residence time will be less. So normally, when the patient is suffering from lung fungal infection, the dose is improved, dose is given more, and that causes a serious problem. When you are giving the doses more, what will happen? Toxicity problem will be there. Toxicity problem will be there. So you cannot run this antifungal drug for long time. That causes, maybe when you are giving a drug with a little bit higher dose, and you have to stop it by 7 or 10 days or 15 days. Otherwise, patient will suffer from severe toxicity. And when you are stopping, the interesting finding is that fungus may die. Fungus in lungs may die, but the spore generated by the fungus, they will not die, they will survive. So as soon as you stop the medicine, maybe within 7 to 10 days, recurrence of the disease will occur. So you see, thereby it is very, very difficult for the people to get rid of fungal infection in lungs. We thought that if we develop, this nanoparticle containing voriconazole, instead of its distribution through blood throughout the body, if you just give this drug or the formulation through the nose to the lungs and distribute it in the lung tissue, what will happen? It will slowly release the drug in locally, and it will produce the local high concentration instead of affecting the entire body. So it will minimize the toxicity and also help to kill the fungus as well as spore, because they're staying in lungs for longer period, maybe few months altogether. Sometimes, it can be there even up to 5 to 6 months, which will be enough to get rid of the fungal infection in lungs, including removal of spores. So, what we have done, we have developed this formulation, and we have studied the drug deposition by using cascade impactor, Anderson cascade impactor, to see that how the drug will be distributed with the size of the nanoparticle, how the particles will be distributed in the entire lung. Because you know that if you want to perform any experiment, you have to get a substantive, convincing data at, at the initial level, just or at the in vitro level, and then you can use it to get the permission for in vivo, and then you can proceed for in vivo. So we wanted to see that whether the drug or nanoparticles containing drug will be distributed throughout the lungs or not. If it is not, what will happen? We have to discard the formulation, because the most important thing is it has to be distributed in all the lobes of the lungs, because human lungs and animal lungs are different. Animal lungs have many lobes, and not only that, that it has to be distributed in all the lungs. Otherwise, if even in one lobe it is not there, the spore will be there, and recurrence of the disease will be there. So that is the purpose, to check whether the deposition of these nanoparticles throughout the lungs will be proper or not. Using the cascade impactor, we have done it first, and once we get that sufficient findings that yes, it will be there in all the lobes, all the areas at the different sizes, we proceed for the study in animals. And in animals, we use this nanoparticle with FITC level. It is, is a fluorescent dye, gives green fluorescence. And you see what we got, a very interesting thing is that it reaches in alveoli. You can see they all reaches in the alveoli. So, this is very important to understand that it must reach the alveoli if we give it through the nasal route, right? Question is, how did we give this to animals by nasal route? This we have published in the IP in 2012. This instrument we have developed. See, this instrument, why we have developed, I will just give you a brief history, brief story. What we did, that in case of the lungs, my student said that I have got two types of ways to deliver the drug into the lungs. One is using the nebulizer, and in the nebulizer, actually, you have to anesthetize the animal. That means you have to you have to make the animal senseless, anesthetize the animals, and uh, you have to treat the animals with this nanoparticle using nebulizer. And the other is you have to fumigate with these nanoparticles in a chamber, closed chamber, in which animals live animal may be kept. I disagree with my students. I say both of them I don't accept. The reason is when we nebulize any people, by the by, by that time we have also brought nebulizer from US to nebulize animals. But I didn't accept this two different plan of experiment. First plan was what? First plan was they saying that we have to you have to anesthetize the animal. You know that when you will find in your house, if somebody is nebulized, you will find in a without senseless condition, he or she gets the nebulizer. So I don't agree that when you are anesthetizing the animal and giving it this nebulizer, this will mimic the exact human condition. So I dropped that idea. And second idea was, in a live animal you are giving it is fantastic, but you are just giving it in a closed chamber, fumigating it entirely. That causes not only the exposure of the lungs, but also the exposure of the entire body. Because animals has lots of hair, hairy body structure, and you know that their skin is 20% more, 20% more, penetrable than human skin. So obviously lots of drugs will go through the skin into the blood. So actual figure you will not be able to understand. So that's why we started thinking and we have developed this. What is this? You just see this is a box. This is a slide washer box. Here we have given to, what you call this uh, wall with this thermocol. So thermocol wall inside, they are having six hollows, one, two, three, four, five, six hollows you can see. And these hollows, we have used this tube, the centrifuge tube. This portion was more open. This side it is there. And the other side is you are putting the animals here. The nose is coming out. You're packing it with this, cotton, and then use this lid so that animal cannot turn, so nose will remain open. So 1113 and 1113. So six animals altogether you can do. So there are two separator, thermocol separator, and there is holes through which you are just putting it, this portion. And there is a small hole through which you are using this dry powder inhaler. We have developed this dry powder inhaler using the same, centrifuge tube of lower volume, and this is this with the plastic pipette. We have made it whole and then joined it with adaldehyde gum, and then you see this is also the pipette you can see, and then you see here is the rubber teat you are using. So you are just when pushing, it will pass through and it will fumigate in the chamber. Nose is here, animal will take the or inhale the nanoparticle fumes through the nose only, body will not be exposed anyway. So you see, through this hole we have taken the picture. You can see these are the nose. You can see of the animals. In test setup looks like this. The animals will be here, 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 here. So the nose are here, and you are fumigating here with these nanoparticles. So they are bound to inhale. So sorry. So this is called nose only apparatus we have developed, and then we thing is we actually applied it to the animal. So basically, you see, for that you don't need any sophisticated instrument, nothing. Only your planning can help you to develop whatever available. You see, using that you can develop your own instrumentation facilities. That is the research. Research does not necessarily mean just I am just swimming in the sea or ocean. It is just what you have planned, thinking, and exactly you have to go to the right location, which can satisfy your hunger of thinking. This is very important. Now, you see, next case. So, what you have seen, that first case we have just giving this using this dry powder inhaler. We found that animals, after the inhalation, it is going to lungs. So, next day, this we published in Molecular Pharmaceutics in 2015. Here you see we use this nanoparticle to see how long it will be there in the lungs. So it is not that it is just going into the what you call is this alveoli is fine. Now we want to see how long it will be there and how much it will be there in the different lobe of the lungs. Here, that major difference is we didn't level it using this FITC, rather we radio leveled it with technetium. So we have used nanoparticle, then leveled it technetium, and then technetium radio leveled nanoparticles have been just, what you call this, through this inhalation chamber we have given to the animals, and what finding is there in the lungs that we will report here. Now you see these are the nanoparticles we have developed. You can see first to picture A and B. This is for SHEM, scanning electron microscopic picture, and this picture ECD, CD and E. They're all you know transmission electron microscopy. Now the difference is scanning electron and transmission electron microscopy is that scanning electrons will give you mostly the surface of this particle. How do they look like? And in case of this transmission electron microscopic picture will give you how it looks like from inside. You see here, if you see the inside, the many black spots are there, the distribution of the dark particles. If you just enlarge it, you will find that there are lots of scattered dark particles are there inside the nanoparticles. So basically, you can get the view of these nanoparticles you have developed. Then we also go for atomic force microscopy that gives you idea about the three-dimensional structure of this nanoparticle. You will get this XYZ, dimension, and then you see once you have just administered the nanoparticles to the animals, what you have seen or what we can see. You see, technetium, its biological half-life is low. So normally 6 hours, so you cannot extend it for a long time. So we have studied it up to 4 hours. That means we have taken the, photography up to the, that is gamma, scintigraphy, you can say scintigraphic image, scintigraphic photographs. We have taken, because it's radio level products, and you see we have taken at 30 minutes, 60 minutes, and 240 minutes. 30 minutes, half an hour, 60 minutes, 1 hour, and then 4 hours, 240 minutes means. So you see, there are two lungs you can see, A and B, A and B in every case. Now, A is basically, you're using nanoparticles containing drug and labeled with technetium. B is free drug, no nanoparticle, but it is also labeled with technetium. But you can see in half an hour it is reaching to the excretory organ. It is reaching to the excretory organ, urinary bladder, and you see in 60 minutes it becomes very less. This portion is gone. So many of these removed. And you see eventually in 240 minutes there is also much greater amount have been excreted. But in case of the lungs delivered with these nanoparticles, you see they're retaining in the lung. So this is also a satisfactory, you know, what you call is this result. Now, question is that is also a qualitative finding that you can see whether it is there or not, how long it is there, but how much it is there. Quantification is the main thing in research. How much it is there, and that we investigated at 2 hours and 4 hours, not only in the lungs but also the different lobes of the lungs. This is very important. The entire lung tissue, and we are given no, it is the different lobes. Animals has different lobes. There are 5 lobes, as you can see, 5 lobes are there. So in the different lobes, all these 5 lobes, how much dark is there? So at 2 hours and 4 hours, you see the presence of drug is there. Although there is a minor variation in the middle, accessory, and caudal, you can say the lobes of these lungs. However, you can see that it is there in all the lobes of the lung. That is the satisfactory findings, because it is important that no lobe should be free of drugs, distribution of these nanoparticles. Otherwise, spore will survive there. So this we wanted to see. Now another study, this is a different study. So there in the first study, what I focus that we have targeted the lungs tissue, but different means, different way. Now, not the conventional way, rather non-conventional way. Here also, this is the presentation of the non-conventional way. Here, we wanted to deliver a drug into the brain, and the drug is docetaxel. Docetaxel is an anti-cancer drug. Basically, we wanted to deliver the drug into the brain. Question is, this time we have used not the nanoparticles, but the nanoliposome. Liposome is not a particle. It is not a solid mass. Particle you understand, like marble. Marble is a particle. Bangla bolle guli. Marble is a particle, solid mass. It's a solid mass. But balloon, you have seen balloon. This is liposome is like balloon. Inside is hollow. Inside could be water. All balloon. You have water balloon. You have seen. So the balloon is basically ballonic structure. Membrane is there, soft membrane, and inside it there is a hollow. So drug can be present in the hollow containing the liquid, or drug can be present in the membrane also. So basically, this lipid nanocarrier, the nanocarrier, but they are basically, liposome. And what we have done, I will discuss this later on, but you remember this particular lipid, DSPE. DSPE is a brain lipid. DSPE, disteryl glycerol phosphatidylethanolamine. So this is a brain lipid. This is very important because we have used this lipid later on. So these are the liposome. Liposome, you see, there normally liposomes are very homogeneous type. Size is homogeneous. We have developed different types of liposome, and you see more or less C, D, E, F. You can see their size is very, very small, less than 100 nanometer, and very evenly distributed. But remember, You can see, if you see it, this is the photo. These are the photographs taken by SEM, scanning electron microscopy. You can see the surface only, nothing else. But truly, it is not particles. So it is very important to go for tame, tame. But this tame, in case of such a small size, just merely tame will not, transmission electron microscopy will not give you enough idea about them. So what you should do, you should go for, cryotame. You have to cut this by pre-fracturing or something like that, and you have to see it under microscope how this liposome is. I said inside is hollow. Now the size, you can see, size is normally is below 100 nanometer is the average size. We have got of different particles have developed actually. So here, this is the cryotame picture. You see, every case lamellarity is there, intact lamellarity. That's why in case of liposome, if you study, it is very important that you should go for this cryotame to understand that there is no fracture, no leakage, nothing is there in the membrane, because that size is very, very small, below 100 nanometer. I think you can see this picture, 37, 38, below even 50 nanometer, the sizes are there. This is also nearly 50 nanometers. So the membrane is intact. This is a membrane thing. Inside is hollow. So by cryotame, you can understand the structure of it. We have labeled it with FITC, and we first wanted to see whether it is reaching in the brain or not. We found they are reaching in the brain. See, one case we have used this normal material. Normal material means phospholipid cholesterol to develop this nanoliposome, and they brought the content is very less in the brain cell, whereas when we have used this DSPE, that is the brain lipid, we found a good amount of drug is going to the brain. This is also the picture taken by gamma scintigraphy, and what you can see, this the animals. This is free drug. This is given by the liposome, by usual liposome, that is phospholipid cholesterol, and this is the liposome containing not only the phospholipid cholesterol but also DSPE. See the difference here. No drug is reaching into the brain. This is a smaller size, larger size of the picture. Here the drug, smaller, larger size of this picture, that is magnification. Here you see the drug is reaching into the brain. Smaller size, you cannot see it properly, but here it is reaching the brain, but very less. But if you see here in these two pictures, and here you can visualize it, it is there. And if you just enlarge the size, you can see it is at least 400 to 500 times more amount of drug is reaching into the brain. What we did when we just tried to see whether it is reaching to the brain or not with this normal liposome, we found it is reaching, but the amount is very less. Amount is very less. You have to maintain the dose, so you have to improve that this liposome containing the drug should reach more into the brain. And for that, we are thinking, how could it be done? We planned that, see, a brain has different phospholipid. Why don't you take some brain lipid or brain phospholipid to incorporate with the formulation so that it can reach into the brain? We did the same, and see the miracle. At least 400 to 500 times more it is reaching into the brain. And that is the way you have to think, you have to plan your research. Research means not copying from others, not cheating from others. Research means your brain should be active. What is the shortage? How you can improve? Always plan from your brain. So next study, also this is the continuation of that study. We have shown, that is the here the plasma level and the brain level of the drug. You can see the when it is the formulation, it is even reaching into the plasma much longer time. After 24 hours, you can see, and in brain also, it is available up to 24 hours, much more. So, in quantity. So, this is, very important. Now, I think, how much time left?
Sir,
Tell me. Yeah.
you have the liberty to take as much time as you
Okay.
want.
Okay. So, next is, you see, I am just going to discuss about the lipid-based nanocarrier, lipid-based nanocarrier, and this is also to deliver the drug across the blood-brain barrier, that is into the brain. But this study is very interesting. We have selected a drug which never, or even if it is, if it crosses very, very low amount, substantially low amount, which cannot be considered as the amount for, treating anything in brain. And that drug is zidovudine. Zidovudine, is a very famous drug used against the HIV. AIDS, against AIDS virus. So problem is, in AIDS virus, it's a notorious virus. Why? Because zidovudine is very successful in against this HIV virus. If you just go for in vitro study, you will find 100% killing of, AIDS virus will be there, but in the system it is not, because they are notorious virus. Why I am telling them notorious virus? Because you know that you want to catch a thief or a terrorist or such a person, and police is just making combing operation throughout the country. But if the person is hiding in the parliament or with the president, it is not possible to find him out. So it is like that. This HIV virus at the initial stage of infection, they hide themselves in the macrophages. Macrophage, that kills the virus, that kills the new infection, new bacteria, whatever invading in our body. But that macrophage, inside the macrophage, they're hiding. Again, they're hiding in the brain. So that's why at the initial stage, it is very important to reach the drug into the brain, so that whatever HIV virus is there in the brain, that can be killed. Zidovudine, I told you, it's a very, very nice drug for killing the virus, HIV virus, but it is not reaching into the brain. So that's why we planned that how we can bring it into the brain in a good amount. Here, very interesting story is that it was a DST funded project. So we thought that we actually working with this normal lipid, and it was not very functioning. So we developed a lipid, changing lots of different combination and ratio, and we found stearic acid, oleic acid, palmitic acid uh, made, this lipid we used, and we compared with the swal acetane, and we found that this lipid is showing at least, even in few percentage, enhanced activity than the normal swal acetane. These are, you can say, the formulations, different formulation. You see, I told you we use this lipid, we use stearic acid, oleic acid, palmitic acid. Ratio we have taken 8.08 is to 4.13 is to 1 ratio, and we made it, and we have named it as a MGF. And then we use this and develop this formulation, and also with the soy lecithin formulation. And here you see we have developed this nanoliposome by scanning electron microscopy. This is with the keratin. Other keratin pictures are here. You can see more or less this is bilamellar, and these are all unilamellar type. You see liposomes we have developed. I think you can see it. Next, also, among other various study, we wanted to see whether it is reaching into the brain or not. And you see, free drug, it is not reaching into the brain at all. Nothing is reaching. Of course, it will not reach, we know, because zidovudine is highly water soluble drug. It is not supposed to reach into the brain. But you see, this is your phospholipid and your newly developed lipid we have just seen. They're very comparable. Even I think it is reaching more as compared to the normal soy lecithin. We have used this MGF lipid, and that was even better. So you see, with a new lipid, we successfully delivered, of course, with the soy lecithin also, we successfully delivered zidovudine into the brain of the animal. Now, if you just go for this, what you called is this pharmacokinetic aspects. Pharmacokinetic aspects, if you see, we use this zidovudine drug, which is commonly known as AZT also. See the half-life of our lipid. I said that it is just going little bit more. So whether it is showing little bit more or less, actual data, you have to produce the quantitative data, and you see in the quantitative data, this is for free drug, so we are not interested about free drug. Obviously, it should have a very low profile of data, so it is not interesting for us. So if we compare these two, it is 9.72 and this is for 7.64. This is the plasma half-life. Nearly there is a difference of 2 hours in case of plasma half-life. This is the half-life. Half-life, you understand. If half-life is more, sustainability of the drug in the blood or wherever, you see, it will be more. So you can see that this is 7 and this is 9. So where is the difference? Just little difference, 2 hours. It is not that. 2 hours difference in half-life is much more. One example, if I give, you will understand that if I say a half-life of a drug is, say, 8 hours or 2 hours. Say if the half-life is 2 hours, after 1 hour, it will be 50% in the body after 2 hours. Then after 4 hours, it will be 25% in the body. After 6 hours, it will be 75%, another 12.5% in the body. So you see, just 2 hours half-life means how long the drug can remain in your body. So 2 hours improvement is also a good amount of improvement. This is whatever I said for the student basically. And also, you see, it is reflecting with the clearance. Clearance data, you see, the clearance is much less. It is 6.73, and here it is, just 5.9 liter per hour per kg body weight. So you see, basically there is also a difference. So that's why I wanted to edge this MGF lipid compared to normal, phospholecithin. So this is also an interesting findings. Yes, this it will take little more time, but of course I have to check from Dr. Roy. If you permit, I will continue, or if you find that time is over, you can tell me, I will leave. So, Dr. Roy, please. Should I continue it?
Yes, sir. Yes, sir.
Okay. So you see, this is now all these three things what I have discussed. They're targeting, but not the what you call this conventional means. They're all non-conventional means. This is conventional means. This time we are using a ligand. This nanoparticles, of course, PLGA nanoparticles containing epigenin. Epigenin, is a very successful flavonoid, and it actually, causes lots of effect, biological effect in the system, including anti-cancer potential. So we worked with epigenin, and actually, it is encapsulated in nanoparticle, and it is conjugated. Nanoparticle surface was conjugated with aptamer. Aptamer, basically, they're oligomers, oligonucleotides, and oligonucleotide, you understand, synthetic oligonucleotide. That means DNA or RNA small sequence, and they actually form a structure, specific structure, three-dimensional structure to bind with certain proteins. So basically, they are called aptamer. We use this aptamer to target the colorectal cancer cells. Here, so this single-stranded oligomer, so naturally the modification was there. We use this phosphorothioate modification so that it can be stabilized in the body. It will not be destroyed by DNAs or RNase enzyme. And this is the nanoparticle containing the drugs, and these are the aptamers just to target. So this is a basic picture. If you just overall study, I can show you that when you are just injecting this aptamer, it is going into the cells because there are some proteins. These proteins with which this aptamer is binding, that is called EpCAM receptor. So EpCAM receptor, we targeted this EpCAM receptor, which are highly expressed in colorectal cancer cells. So, and then we have added it with this, what you call is this aptamer. Aptamer will bind with this nanoparticles, aptamer, and this EpCAM, they bind and they go inside the cells. Then, it is with this endocytos, what you call this endosomal escape is there, and then it's coming out, and from there, the drugs are actually distributing in the system, and it works to kill the or destroy the molecular mechanism of the cells to induce it towards the apoptosis. So that is basically the thing. Here we have developed this aptamer conjugated nanoparticle. This picture you can see, this is aptamer conjugated nanoparticle at the well, because they're heavier, so they could not move. This is the free aptamer. It is moved here. You can see this is the nanoparticle without any aptamer. That's why you have not seen any white thing, and these are the ladder. So what we have seen, that aptamer conjugation with nanoparticle is there, and that convenes us, and that is the usual FTIR. Then just to understand the any drug chemical interaction, and then you see these are the nanoparticles, these are the size, and zeta potential we have mentioned, because these are the very important physical data to understand their size, range of size, and also how its surface charge is there, because surface charge depends. Based on the surface charge, we can decide that whether it will remain in the suspension for longer time or shorter time. And then, of course, these are tame. Here you see the drug is solubilized. That's why you see it is entire particle is black. Otherwise, it could have been scattered with black spot. And this is your what you call this AFM. You can see the surface, and also the drug release pattern from the sustained drug release pattern from the nanoparticle. Aptamer conjugated and non-conjugated. There is a very almost hardly any difference between these two. That means aptamer conjugation actually could not vary the drug release pattern much in terms of or from the nanoparticles. Now, if we just go for these cells, colorectal cancer cells, we have taken HCT116 cells, and here you see it is FITC level nanoparticle, it is there, and of course we found it is in terms of facts, we have quantified it, and we have found that with the times, the study has been done from different hour time points, like 2 hours, 4 hours, and 6 hours, and 24 hours. And so accordingly, you see the variation through the facts result. You can see the drug, although this facts result we have taken up to 6 hours, And here, in every case, you see the drug content increases into the cells. Once they're reaching into the cells, we have also tried to see whether it is with this technetium level, whether it is reaching into the colon or not. Of course, it is they're reaching into the colon, we can find. And then we have just isolated those portion where radio level is there. We have taken those tissues and then we have studied with this, what you call this histopathology, and just see whether they're the normal tissue or cancerous tissue. And also we did it with this FITC level, and there also we have found that FITC level formulations reach to those tissues in other animal wherever it is going. We have taken those portion also for study, and you see these areas at different hours of treatment. We have given you this 2 hours, and this is 5 hours in case of controlled. This is 2 hours, 5 hours, and this is DAPI, and this is the March figure. So it is going into the tissue. There is no problem about that. We can understand it. Now, important issue is that it is going into the tissue. We have quantified it. Also, it is going into the cells. In case of this FACS earlier study, that is this study, you have seen it is going into the cells, and with this FACS, we have quantified it. With the time, it is increasing, and also it is going into the tissue. We have found with this experiment. Now, what we want to know that at different hours, basically, what is its impact with the mitochondrial membrane potential. Now, here I want to tell you that it is done with the JC1 staining. JC1 staining, you see that mitochondria which are actually lost their membrane potential, they remain monomer. They remain monomer. But the active mitochondria, which they're very active, they actually conglomerate. They conglomerate, and they become, you can say, that aggregates, and that aggregates become red in color. So aggregates under the confocal microscopy or microscopy, you will find red in color, and the monomers you will find green in color. Although here it is the color selection was blue, because that is, you have the option by fax, you can select the color, but actual case you will find it is red and blue. I beg your pardon, green. So this green color, you see here, you see the lower portion is always green and the upper portion always remains red. So you see here the, if we just find that in the tissue there is a huge damage of the mitochondrial membrane potential with the times, and that indicates the cells are in the post process of death up to, we have studied it up to 48 hours. So in 12 hours and then up to 48 hours, the studies are there, and we have found that the damage is much more in case of the, that is what you call this mitochondrial loss of membrane potential is much more than the aggregates. So that is a good indication that cells are dying. Also, we have studied the different enzymes in those tissues, like, as you see, basically we have studied with SDH and also glutathione level and NADH. So here you see the GSH, that is reduced glutathione, is reducing, and this oxidative enzyme is increasing. This is also reducing. That indicates that there is a huge oxidative stress into the cells that actually navigate towards the killing process, apoptosis, and that also damages the mitochondrial membrane potential, which reflects the, what you call this apoptotic process, because they are very much correlated. Mitochondrial loss of mitochondrial membrane potential means, there will be the apoptosis. We have seen in the tissue just before and after treatment, you see this is normal. These C, D, E, F are cancerous, and this is after the treatment. You see the C, D, E, F, you can see the lots of damages in the cells, and you see after the treatment, the improvement is there, much more similar, more particularly I can say J variety is more similar to the normal B variety. So there are some improvement after the treatment of aftermark conjugated nanoparticle, which is not much there in case of usual nanoparticles we can see. That could be the non-availability of the drug or the formulation to the right site and also for longer period. Now, this presentation, I will not take much time. I understand that those who are listening, they will also be saturated. This presentation, possibly the last presentation, this is very interesting presentation. That's why I have just selected that peripheral nerve targeting. This study has a long history. You know that the one seminar I went to present my work. This was in Beleghata ID Hospital, and there a doctor came and they said that can you just target the peripheral nerve using one drug I will give you. I said which drug? He said ribavirin. Ribavirin is a very costly antiviral drug. So ribavirin basically it acts very nicely against the rabies virus in vitro, but in vivo it doesn't work. The reason is that in vivo the rabies virus they resides in the peripheral nerve, and many drugs actually do not reach to the peripheral nerve. So he said, and then we came and I said, you send me the drug. Within 7 days, he sent me the drug. It's a very costly drug. The company actually came and gave me the drug. And then what happened, that we started planning how to do it. I planned that why don't you we use procaine to conjugate with the formulation so that we can bring it easily to the nerve. We use procaine actually conjugated with this drug, the formulation containing ribavirin to target the peripheral nerve, because procaine, it goes to the peripheral nerve. We are all 100% sure about it. Now, I have given it to the first group of students to work with for 2 years. They worked just for the sake of conjugation with the surface procaine. They were not successful. Then in the second group of students, it took 2 years. Then another group came and he worked. I spent nearly 1.5 lakhs for this conjugation process. This was lost again. So after 4 years, one postdoc lady came to me and I asked, and finally, after 3 to 4 years, she was successful. And thereby, you see, this procaine we have conjugated with the formulation. We developed the liposome, and that liposome we have ejected, and it worked. So, how we have conjugated? This is the description of it. That this is the method we have adopted. Procaine conjugated. It took nearly, I said, 6 years, and then another 2 years, 2 to 3 years we used for this experiments, biological experiments. Finally, after 10 to 11 years, we published it. So, what is there? This conjugation, successful conjugation process is there with NMR data and mass. Then you see, these are the formulation we have developed, and this is the liposome. You see, uniform size and uniform lamellarity is there, no leakage, nothing. And then after that, we used it is the drug release pattern, and we use this for the animal investigation. We labeled this formulation with technetium. What we found that it is reaching into the muscles. It is reaching into the muscles, technetium. Now, when we just use this for publication, editor said, okay, I agree, it is reaching to the muscle. Can you say that it is reaching to the nerve? Do you have any other proof to show that it is reaching to the nerve? Because you are targeting the peripheral nerve. Then what we actually use myogram. And you see, here this is the same scale. You can see same scale. There is no variation of this scale, same scale. And this is your, we have used this normal animal, and this is the treated animal. You see the impulse level of nerve is much less here. That indicates definitely procaine is reaching there. That's why the nerve impulse is reducing. And that has been accepted by the editor, and the paper was published. So you see, thereby, you see, we have successfully targeted the peripheral nerve also. So I have showed you two things. First three experiment I have showed that without the conventional targeting, we have targeted, and using the conventional targeting, last two, aptamer and this procaine, we can also target the uh, affected site. Now, I told you that I will also speak few sentences regarding the nano hazards. Nano hazards means nanomaterial is always not safe. You see, it could be a saver, it could be a killer, like formulation, what we have mentioned. They can save you against cancer, they can save you against viral diseases, they can save you against many other diseases. However, when these nanoparticles are coming through the as pollutant, as you see, that can be detrimental for your health. A nanoparticle thus can save your life or even can kill you by producing cancers or this thing. So you have to be very careful that nanoparticles are roaming around more as pollutant, less as drug. So these are the things, whatever I have mentioned, this is related to nanoparticle hazards. I have published this work, two works. Those who are interested to work with these nanoparticle hazards, they can go through these articles, and this will be good for you to understand it. Now, basically, nanoparticle is a costly affair. Nanoformulation is a costly affair. It should not be a burden for all of us. You have to make it in such a way that someday or other it will be less costly and available for many of us. So, of course, the efforts are required for that. Anything you want, anything I want cannot be successful immediately, but efforts are required. If there is a will, there is a way. You see, this goat, so many plants, so many leaves are there, but it was interested to take plants from this tree, from the treetop. It reached and taking. So efforts are required. If we have the substantial effort, we'll be successful. Thank you very much. If you have any question, queries, please feel free to ask.
Okay, I truly appreciate
So
you,
I
sir,
want
and sharing your valuable time with you and spent helping us as well as all the faculty members present out here. Thank you very much, sir. So now I'd like to request all the faculties, like whether they got any queries regarding the topic, you can raise your hand.
Can I escape this presentation mode?
Sh- Sure, sir. Sure, sir.
Okay, because so that I can at least see all people. Yes.
Yes, yes.
If
So
anybody is
anyone from the
having any queries,
faculty,
yes. Yes.
so anyone from the faculties, do you have any queries? Please raise your hand.
Or if you want to know anything, or if you have a kind of thinking which you want to get it clear, you please feel free to ask me. Don't worry about anything.
I shall be requesting the faculty members to be ready with your questions and to enrich your knowledge. Your queries will be enriching your knowledge only. I should request to all of the faculty members again with your questions to enrich your knowledge.
How many of you did not understand what I have said? Raise your hand. So anybody, if any question is there, please feel free to ask me. Otherwise, I will quit.
Sir, I want to ask you one question,
Yeah,
sir.
please feel free.
Just to enrich my knowledge. Sir, please guide me how to penetrate the blood-brain barrier for any nanoparticle.
Easiest way, I think easiest way to cross the blood-brain barrier is prepare nano-sized liposome. This is exceptionally very good. If you find that it is reaching but the concentration level is low, in that or drug amount is reaching low, in that case you just use this any brain lipid. It will be exceptional. Basically, you can use DSP because these are all authenticate and already we found it is correct.
Thank you, sir.
Yeah, nanoparticle also can work, but nanoparticle size should be small, maybe below 100 nanometer or within 100 nanometer, not more than that. And second thing is it should be lipid based, like it could be PLG is lipid, or any other lipid based.
Hmm.
Okay.
Sir,
Okay.
which one shall be better? Well, mean we are trying to formulate a nanoparticle or we are trying to formulate a nanomycel?
If you ask me, I will prefer nano liposome
Okay. Okay, sir. Thank you, sir.
because this is very successful to reach across the blood-brain barrier.
Okay. Okay, sir. So, any other queries from any other faculty members? I just wanted to say that.
Yes.
Hello,
Yeah, somebody is going to ask.
sir. I had one question, sir. Sir, what is the significance of using, sir? That is for surface microscopy. That is we're using the, sir, transmission microscopes and a scanning microscope. Sir, I'm asking the what is the significance here that we used, sir, scanning microscopy for studying the surface.
Scanning microscope actually provides you the idea about the surface.
Okay,
But
sir.
let me tell you that another
Hmm.
variety is there. Field emission scanning electron
Okay.
microscopy. That
Okay,
gives
sir.
even the three-dimensional structure. Okay. So if you
Okay, sir.
go for
Okay.
FESEM, you will get the
Hmm.
three-dimensional structure as well as
Hmm.
surface. But if
Okay.
you just go for this, only SEM, you will get the surface structure.
Hmm.
Surface structure give
Hmm.
you the idea whether the surface is smooth, whether the
Hmm.
surface is having porosity, whether the surface is tortuous, whether surface is having some leakage. So everything you will come to know. So surface investigation is very important. And secondly, SCI, 10
Hmm.
transmission electron microscopy will give you the idea how it looks like or what is there inside this.
Yes, sir.
So one thing is you are just observing a three-dimensional thing from the surface, and
Hmm.
another is what is there inside. Okay, so
Oh, okay, sir.
that's why these studies are important.
Any other queries? Any other queries?
Sir, I think there is no queries for our faculty groups. So, sir,
So,
let's.
one thing that listening to BM sir is
Okay,
like
Dr. Ranu is here. Madam,
Yeah,
would you like
please.
to ask anything? Dr. Ranu, ma'am, would you like to ask anything? Ma'am, you are
Audible?
not. Yes, yes, yes. You are audible. Please, ma'am, carry on.
Sir, you told for targeting to the brain crossing the blood-brain barrier, we have to use the DSP lipid. As I know, the soy
It's
lecithin
not
is
that
already
we
very.
have to use. We have to use it, but this is a great option to use because we were very successful using that. That I said.
Ha,
Right?
DSP.
Yes.
If we use this lipid, then we will get more concentration in the brain.
Hmm.
That is experimentally proved, as you
Hmm.
have shown.
Hmm.
But as we know, the soy lecithin is already costly, and how much the costly will be DSP lipid? So, is it possible for a common?
You see, it is not that costly. It is not that costly because these are
It
available.
it is cheaper than the soy lecithin.
Maybe the like you can say little bit variation is there, but not that very substantially expensive. Not like that. Because
Okay, then
these
okay, sir.
lipids are available and they synthetically they develop it nowadays.
Sir, one thing more, you have experimentally proved that that lipid, stearic acid, palmitic acid,
Yeah,
and oleic acid, a particular combination, you have developed that lipid for
yeah,
cross preparation of that nanoliposomes.
yeah.
May we use this combination of the lipid for development of the liposome for targeting to the other part of the body?
That we have not tried. People can try because this was a DVT project. It has a history actually. That history I did not disclose because we didn't have much time for that. But yes, we have tried for this because it was a DVT funded project and it was mainly intended to deliver the drug across the brain. So we tried it that. But other organ we have not tried. But I think it will be successful. Why not? Because if it is successful to reach in brain, of course it will reach in other organs too.
If it is possible, sir, then I think it will be cheaper than the lecithin because lecithin is too much costly.
Yeah, lecithin purpose is, it is costly. Everything is costly. If you prepare something, and that will be always cheaper. But thing is that cost effectiveness. I do not know that when you are developing it, whether it will be less costly or more costly. Because when you are developing a lipid, you are using several combination. You are testing and you are ultimately selecting that also cost more. Like if you get it in ready in hand, that is different. But when you are developing it with the lots of experimentation you are doing, that costs more.
Initially, that will be
Like
expensive.
if you just say, if you want to get the lipid from me and I'm giving you, it is less costly. But when you are developing that, it costs more. That I wanted to tell.
Yes, but once it has been developed further, if we will use that one in that regard, that will be
That
cost
way
effective.
maybe it is less. But we have
Cost
never
effective.
calculated the cost, how much
Okay,
it is
okay.
coming. I believe it will be
Thank you,
less.
sir. Thank you, sir.
Okay, so thank you so much, sir. I enjoyed every minute of your lecture as well as your marvelous sense of humor. And please give my best regard to your laptop because it it did so much. Principal sir, would you like to say something? Please unmute yourself, sir. Sir, please unmute yourself.
I was just trying to say that listening to BMS's lecture is to gather the particles from the seashore in front of a huge sea, in front of the
Yes,
huge ocean. Just listening to the lectures will be able to make you eligible to pick up the particles from the seashore in front of the huge ocean. I
yes,
was a
sir.
witness.
Indeed.
Of, I know that when I was in JU, sir used to teach to the students from Latchman and from Alton without taking the book in the classrooms. So you can easily know that what's an ocean of knowledge he's having inside him. So no questions will be there for sir's lecture. Only to listen, and only to listen, and make
No, no,
yourself.
no.
Yes,
That is
yes, sir.
exaggerated. Rather, I feel proud if somebody asks some questions because I always I am just always fond of answering people or clarify the things.
So we fortunate that sir, we had you on the first day of our FTP. No person in this country can become Dr. Biswajit Mukherjee. No
No,
person can do
absolutely, sir. Absolutely, sir. Absolutely.
that. I know that his door is always open for your queries. Whenever you will knock your door, his door will be open for you to deliver any bit particles of the sea, any droplets from the ocean. So with that, I'm just giving the turn to Samadeep, to close the ceremony.
Yes, sir.
And from my side, I wish, sir, I just deliver a very big thanks, which is very small enough from my side to deliver to sir, and to make the first day of the FTP a huge success one.
Yes, sir. Thank
Sam,
you so
please.
much, sir. So to be very honest, I enjoyed every minute of your lecture, as well as I think all of the participants out here, they have enjoyed it a lot. Okay, and just because you got a marvelous sense of humor, and please, I'd like to say one thing: you are a huge person. You comes under the 2% scientist of India. So that's also, sir. Let's stop our program out here. So it was nice having you, sir, along with us. It was a small faculty developed program that has been started from today. Okay, and sir, you are the first one to give us a marvelous lecture that has been started with you. So here we are discontinuing the program over here. Thank you so much, sir. Thanks
Okay,
for being a
thank
part of
you.
this. Thank you so
Thank
much.
you all,
Thank
and
you,
wish
everyone.
you the very
Thank you all the
successful
participants.
Thank you,
FD.
sir.
Thank you.
Thank you. Okay, then,
Thank
bye
you,
bye.
sir.
Okay, thank you. Bye bye. Good night.
Sir.
Bye, sir.
Bye.
Thank you, sir.